Hello, today we're going to talk about gene expression. Pretty much, cells contain mechanisms that allow them to express their genetic information selectively, allowing them to turn genes on and off. It's very important for the functioning of the organism to have that control. So, gene regulation is the turning on and off of genes, pretty much turning on and off transcription. All right, this allows organisms to respond to environmental changes. It could be within the chemical environment. Maybe certain digestive enzymes that function in the intestine are only turned on when there's food in the intestine. Or maybe it's the physical environment. Fluctuations in pH, temperature, moisture, photoperiod can change or trigger changes in the gene expression that would occur. It also could be in response to hormones. Many hormones trigger metabolic or structural changes in target cells when they require activation or suppression of gene activity. Um, Pretty much in 1960s, biologists had a clear understanding of this gene expression in prokaryotes, in particular with E. coli. It really wasn't until the 80s that they had an clear expression understanding of that with the E. coli. So let's just talk about the E. coli and lactose, okay? E. coli uses three enzymes to break down lactose. Remember, lactose is a disaccharide. It's made up of glucose and galactose. There are two control sequences that are located next to the lactase enzymes, okay? So the promoter is the site where RNA polymerase attaches and initiates transcription, where the operator acts as a switch which determines whether RNA polymerase can attach to the promoter and start So when you ingest lactose, you need to break it down with enzymes, okay, so you can get the energy from it. If lactose is not present, you don't want this enzyme turned on because you don't want to waste energy producing the enzymes necessary to break it down if it's not present. Okay? And this is just showing you the promoter again, which is where RNA polymerase is going to attach, and the operator, which is the switch that determines whether it can attach the RNA polymerase. Now, an operon this is for the prokaryotic cells. This is a cluster of genes with related functions, the promoter and the operator. Again, this is only going to be for the operon, and this is kind of the switch that turns on and off. It's a functional complex that contains the genes that contain information for making those enzymes of that pathway. And it's all clustered together on the chromosomes. And it is, all of the genes of that operon are kind of the functional unit. Okay? Okay. So a repressor is a protein that binds the operator to block the attachment of RNA polymerase. That will turn off the transcription. It physically blocks the attachment, so it will not occur. So with this, lactose will interfere with the attachment of the lac repressor. That would change the shape, can't bind to the operator, and then the operator switch will be on. Activators are proteins that turn operons on by binding to the DNA. And it makes it much easier for the RNA polymerase to bind to the promoter. And that's going to allow the bacteria to survive in the frequently changing environments that they have. Okay, so again, all of this was for the lac operon with the prokaryotics. Now we're going to focus a little bit on eukaryotes. And again, this wasn't found out until after the prokaryotic expression was occurred. So each eukaryotic gene has its own promoter and other control sequences. Pretty much the default state for the genes is going to be off. Okay, we have more genes, we have more proteins, so the default stage of off is more effective. Okay, and again, this is a little bit more elaborate. So we have transcription factors. These are the proteins that assist eukaryotic RNA polymerase from binding. Okay, the activators are an example of one. The enhancers are usually farther away from the gene. This leads to binding of the DNA that leads to the interaction with transcription factors. Okay, so this regulates the gene just like the operators did. And then the silencers are the ones that inhibit or stop the start of transcription. Okay, so if you have any questions, just let me know and we'll talk about it in class. Thanks.